Harletan. Good morning. I think it's our third day of conference. It's our last section, but not least. It's one of the most important one. I think that's where our young scientists will present their projects, and I think it's very important to see, for our young researchers as PhD students or master students to present their work, and that will be exciting to learn how they perform, not just in a lab, but how they can present their projects. I think it's very critical for their training, and this is really important. And considering how important that section is, so we also have very important speaker to start this section, and our Vice Dean for research of the School of Medicine, Dr. Dieter Reitmacher, will present our important speaker right now. Thank you. Please.
Okay, thank you, Dos, for introducing me, and now it's my pleasure to introduce our speaker. It's Thomas Jentsch, and actually I know Thomas for more than 25 years. And as Dos already indicated, it's the day of the young scientists, so some of you guys were probably just born when we already met and were doing research. So I want to briefly introduce him. I don't go through the details, you have everything in the booklet, but I want to mention clearly that he is internationally recognized for pioneering the discoveries of ion channel research, and definitely the chloride channel family, where he became famous for not just in Germany but across the globe. He won numerous prizes, and I just want to mention the Gottfried Wilhelm Leibniz Prize, which is the biggest research prize you can win in Germany, the Prix Louis Jantet de Médecine, so French prize, Ernst Young Prize, just to name a few, and I'm very much looking forward to his presentation today. If I'm not mistaken, this is based on some recent results that you published in Nature Communications just this year. So welcome here on stage for this keynote speak.
Okay, thank you very much, Dieter, for this kind introduction. Indeed, we met in Hamburg in 2000-something, a quarter of a century ago, more or less. And I'm really pleased to be here to continue a little bit on my talk I gave yesterday at the NUAC, in which I made a general introduction, more general introduction into chloride channels. And today I will focus on our latest research in this field, but I will also give you a short introduction, of course. And it's an honor to be in the session of young scientists, even though I have progressed a little bit from being a young scientist. Okay, so my talk will be about two new beta subunits we have discovered for transporters in intracellular organelles, and it doesn't progress. Maybe I have to turn it on. Okay. Okay. So we are talking here about endosomes and lysosomes, which are derived from vesicles pinching off from the plasma membrane, and they, of course, take up many different substances. And as you know, you can distinguish early endosomes, sorting endosomes, and recycling endosomes. And the sorting endosomes can also end up in late endosomes and finally go to lysosomes, where many materials are degraded by lysosomal enzymes. And it is important to realize that the ionic composition of these compartments really changes over the course of maturation of these vesicles. Initially, the vesicles have a pH of about 6.5 or so in early endosomes, and the further you get down to lysosomes, the more acidic it gets until you have pH 4.5. And this acidification is carried out by a proton pump and H+ ATPase, which is located on these vesicles and part with different molecular compositions. But they also have these two chloride H+ exchanges of the CSC family, a family of chloride channels and transporters we discovered in 1990, that is in the last millennium. And I would like to emphasize that these transporters have different functions for these vesicles. On the one hand, they are important to neutralize the currents of the H+ ATPase, because the H+ ATPase pumps positive charges into the endosomes, and this has to be compensated. Otherwise, you would just get a change in voltage and no net transport of acid equivalents. So it's important for the acidification, but these transporters also accumulate chloride into this vesicle. So if you have an acidic compartment with high H+ concentration, the countertransport will lead to a secondary uptake of chloride. And both the pH and the chloride are important for the functioning of these compartments. For instance, lysosomal enzymes depend both on an acidic pH and many also on chloride, and also the trafficking of these vesicles depend on the ion concentration in the vesicles. Well, we have studied all these transporters and many others, of course, and did also knockout mouse models for them, for all of them, and we and others discovered diseases which are due to mutations in these transporters, and these range from impaired renal endocytosis and kidney stones for CSC5, which is highly expressed in the kidney, over intellectual disability, neurodevelopmental disorders, neurodegenerative disorders, in part very severe diseases. And with the lysosomal one, you also get osteopetrosis as a main symptom, that is too thick bones, in addition to lysosomal storage in the CNS. So these are really important for the function of the organism as a whole. Just one example here to give you a little bit biological background. There's one of these transporters, CSC3, which is ubiquitously expressed in every cell in the organism. Initially, we didn't know what it was good for, so we knocked it out in mice, and the phenotype of the mice was a very severe neurodegeneration. So after a couple of months, the mice really have big holes in the brain, where normally the hippocampus is, and at the left you see the progressive degeneration of cells in the hippocampus in the brain. So it's a very severe neurodegeneration. It comes also with retinal degeneration, so these mice are also blind, and they're systemically sick. They are smaller and do not grow as well over time. Only recently, that is 3 or 4 years ago, we found together with human geneticists that CSC3 is also mutated in humans. So there was a lag from the mouse model to the human disease of about 20 years or so, even though we looked a little bit for mutation in CSC3. So it comes with neurodevelopmental and neurodegenerative disorders, facial abnormalities, differences in the MRT, and we even have the equivalent of the mouse knockout in humans with a stop codon on both alleles, homozygous. Here the hippocampus is still there, but it's thinner, and it's the most severe case with CSC3 mutations, as you would expect. Well, now we know that there are many different, not too many, but several different mutations in CSC3 in humans. For the other CSCs, we have many more. So for CSC1 with myotonia, we have about 50 to 100, and so on mutations in these genes, and these are just the mutations in CSC3. Well, this is now an introduction to the entire CSC family of chloride channels and transporters. We identified them by cloning a chloride channel from an electric fish, the torpedo, and this gave the entry point by homology screening at first in a wet lab, and then, of course, also by analyzing the genomic databases into the family of these channels and transporters. And these come in two different flavors. The first group, CC1 to CCK, are chloride channels at the plasma membrane. And they have different functions. CC1 is expressed mainly in skeletal muscle, and when it is mutated in a mouse model at first, shown by us, and then in humans with many different mutations, you get myotonia, which is muscle stiffness. People cannot relax easily the muscle once it is contracted because the muscle is hyperexcitable. So the chloride channel stabilizes the memory potential a little bit like a potassium concentration in most other cells. CC2 is broadly expressed, and there we have phenotypes like leukodystrophy, in which the white matter of the brain shows vacuoles, also in humans, first seen in mice by us, and the activating mutations which cause hyperaldosteronism, that is very high blood pressure. I don't go into the mechanism, I showed it yesterday. We have CCK channels, which are mostly expressed in the kidney. They're involved in transepithelial transport in the kidney and in the inner ear. And the mice and the humans have a severe thought loss in the kidney and are deaf because you need transepithelial transport in the inner ear to have the right environment, ionic environment for the sensory hair cells. They need a positive potential extracellularly and high potassium concentration. But here I'm focusing more on the other members of this family, 5 different members, and these are all intracellular, as I've shown you on these vesicles, on endosomes and lysosomes. And these transporters are not chloride channels, but rather surprisingly, chloride H+ exchangers. So they're transporters exchanging chloride for protons, 2 chloride for 1 proton. Even though the structure is pretty similar, and it's not really understood in detail, even though we have cryo-EM structures and so on, how this difference really comes about. And here again, I showed you in a short summary, we have all these different diseases. These chloride channels and transporters also come with some beta subunits, that is accessory proteins, which come together with the real transporter protein. And we have, when we began these studies, necessary subunits called Bartin for the CCK channels and OSTIM1 for CSC7, which are necessary for the stability of the protein transport to the plasma membrane or to the lysosomes, and also for the function of these channels. And only recently, and this was published this year in 2 papers in Nature Communication and Nature Structural Molecular Biology, we identified 2 new subunits, TMM9 and TMM9B, which we shortly call T9A and T9B, for all 3 of these homologous proteins. And I will tell you the story about these new subunits and their function. This started with our collaborator, Bernd Fackler at the University of Freiburg, using a proteomic study. So he isolates brain membranes and separates the brain membrane proteins in native blue gels at very high resolution. He freezes the gels and cuts them into very fine slices. And for each of these slices, he does a whole proteome. And therefore, when you look afterwards for the peptides, you can make virtual Western blots, if you like, for any of the proteins which are detectable in the proteome. And this is, of course, very handy. And because it is blue native page, you see which proteins really co-migrate. And he realized that CC3, 4, and 5 co-migrate with 2 small proteins, T9A and T9B, in a complex. And there's no other significant peak in the blue native page for these transporters or for the beta subunits. So more than 90% of these proteins are really associated in a single complex. And they, and in my lab, we also did CoIPs, classical CoIPs, which are displayed here in a not so usual Tisny plot. And we see that they form complexes CSC4, 5 with T9b, and T9a. Well, what are these proteins? A little bit the basics of these proteins. These are type 1 transmembrane proteins with an N-terminus in the lumen, or extracellular, if you think of the plasma membrane. They are quite homologous to each other, some 60% or so, a very small gene family. They are homologous in the extracellular or luminal domain and in the transmembrane domain. In the C-terminus, not so very much initially, but in the extreme C-terminus, also very high homology. And these proteins are found in all animals, and mammals have two isoforms, T9a and T9b, but it's really found down to C. anemona, C. elegans, Drosophila, and so on, so it's evolutionarily conserved. These proteins against which we generated antibodies are found all across the tissues, which coincides with the broad expression pattern of the CSCs, and they are localizing to endosomes, which corresponds to the endosomal localization of CSC3, 4, and 5. We also generated pretty early on knockout mice for both isoforms. These knockout mice are viable and seem to be happy, but when you cross the TMM9A knockout with the B knockout, we do not get mice. We got 1 or 2 mice, but they die very early on. So they are really essential for life. And we also looked for the abundance of the proteins in these knockout mice. I don't show you all these Western blots, which were done by Rosa here. So we see changes in the abundance of the respective proteins. So when you knock out T9b, T9a is going up. We also see changes in the abundance of CSC3 in the knockout of A and heterozygous for B and so on. We cannot check, of course, for the double knockout because it's not living. But we also looked at fibroblasts from the double knockout mice. And here we see that CSC3, 4, and 5 are pretty much gone when you knock out both subunits. So the stability of the CSC proteins depend very much on the expression of their beta subunits, showing that they are really essential. And CSC7, a nice control, which is the lysosomal one, is not affected as a control. Also vice versa, when we look at our knockout mice for CSC3, 4, and 5, we have all these mice, we see changes in the abundance, for instance, here for T9a, when you knock out CSC3, or also for T9b and so on. So the stability depends on each other, and also the glycosylation depends on the partner protein, suggesting that in part they're responsible for the R2 transport. But what is now the physiological function of these beta subunits? So we expressed one of these transporters here in xenopus oocytes and frog oocytes. This is now CSC5. CSC5 is in early endosomes, and it goes in part to the plasma membrane, so you can study it by classical electrophysiological methods. And here you see the currents of CSC5. I have to explain it a little bit. These are currents at different voltages, which are superimposed in time here. So the most high current here is obtained at plus 100 millivolts and plus 80, plus 40, and so on. So this is a typical CSC5 current. It is strongly voltage dependent, being seen only at positive voltages. And when you co-express with the beta subunits, the current is pretty much gone. So these are inhibitory subunits in some respect. And of course, you might argue there can be at least 2 different effects that the currents are gone. One is that the transporter does not transport anymore, no current, or that it doesn't come to the surface of the oocyte and you cannot just measure it. So we inserted an extracellular or luminal epitope into CSC5 in one of these loops, have an antibody binding to it, and a luminescence assay to determine the surface expression. And you see this is the surface expression of CSC5. And when we co-express with either 9a or 9b, the surface expression is very much gone. So this could explain the lack of cards we are seeing here. But of course, this does not exclude that there's an additional inhibition of cards. Well, then we try to find out which are the segments of the proteins which really do the trick, which are responsible for the effect. When you look for trafficking, of course, you look at cytoplasmic regions which might bind to sorting molecules, and we did a number of deletions in the C-terminus. And when we deleted, shown in red here, pretty much the entire C-terminus, the transmembrane domain is underlined here, then we do get surface expression of the H8-tagged CSC5 and also of the H8-tagged T9b, and we also get carns. But when we make the smaller deletion here, we do get surface expression in both cases, but we lack carns. So this indicates that this segment over here, the blue segment, is responsible for the sorting endocytosis maybe from the plasma membrane of the protein, but that there might be other things hidden in the C-terminus. So we looked for an endocytic signal here, but this sequence is not conserved between 9A and 9B and across the species. So at first we were a little bit confused, but then we saw that there are a lot of negative charges, aspartates or glutamates, and this is reminiscent of a so-called acidic cluster motif, which is not very well known, which is involved in trans-Golgi sorting and also in endocytosis from the plasma membrane. And you see here across the species down to C. anemona, you find negative charges in the segment here, but the sequence is not conserved. So at first it was difficult to recognize. And these acidic clusters come also in general with phosphorylatable amino acids like serines and threonines or also tyrosines. And it has been shown for other systems that phosphorylation of these residues, which introduces more negative charge, is important for the functioning of these motifs. So we went ahead, of course, and eliminated the negative charges with alanines, and then you get really surface expression in both cases here with 9A and 9B. We mutated also some of these phosphorylatable amino acids, and indeed, these are phosphorylated in proteomic databases. When phosphorylation is studied, you find them phosphorylated, and also our collaborators in Freiburg by mass spectroscopy found them phosphorylated. So we believe strongly that there is an endocytic signal which can be further modified by phosphorylation, so the trafficking might be really regulated by phosphorylation. The same surface expression can, of course, also be detected when you transfect HeLa cells. In this case, you really get it by immunohistochemistry to the surface when you destroy this motif here, the negative charges. So we have this endocytic motif, but we still have inhibition of cards when we get it to the surface. So this indicates that the C-terminus might be important for inhibiting the cards, the ion transport itself. And we went ahead, of course, to do mutagenesis here, doing alanine scanning. You see it over there. When we make a very large deletion of the C-terminus, we together always with a mutation in the acidic cluster to get it to the surface. So you have to do the two mutations at the same time. So you get these cards. And also when you go down and do smaller deletions, you do get it to the surface, but to a slightly less degree. And this indicates that the binding is not really limited to a certain small region of the protein of the C-terminus, but is distributed over a long distance, so that a large part of the protein binds to some part of the transporter protein. And I think this is also indicating that you might have synergistic effects when you have phosphorylation here and there. Then you can synergistically weaken the interaction and activate the channel as a transporter, sorry. Well, to study this in more detail, we established a vacuolization assay because these studies have been done always at the surface, at the plasma membrane, which is not the real important physiological environment for these endosomal transporters. So we looked at vesicles where these transporters are normally located, and we established a swirling assay for the swirling of these endosomal vesicles. And this has been described previously for CSC3. When you transfect CSC3 by itself in cells, you get large vesicles, which is shown here. They really blow up these vesicles. And when we co-transfect now, this is only seen with CSC3 and not with CSC4 and CSC5. We'll come to the difference later. So we decided to study it in the framework of these vesicles, and this is very much supported by evidence from others and from ourselves that this amplification or the swelling of the vesicles depends on the ion transport. There are two types of mutations you can do in these transporters, a so-called UNC mutant, which uncouples the chloride from H+ transport, so you convert it from exchanger into a channel, or you have a transport-deficient mutant, which totally abolishes transport. And this abolishes the formation of these large vesicles. So formation of large vesicles depends on the transport activity, because the transporter, when it is hyperactive, accumulates chloride, and there's osmotic swirling and less pinching of vesicles to make the vesicles again small. Here we have used also a dye for acidic compartments, lysosensor, to make it more evident, and we used it also in FACS experiments to really look at the expression of these large vesicles. Importantly, this swelling is inhibited by T9A and T9B, so we show that the inhibition is not only at the plasma membrane, but also in the intracellular organelles. And this is important because the lipid composition, of course, is different. For instance, at the plasma membrane, you have PI4,5P2, and in lysosomes, you have PI3,5P2, different lipids which have different regulatory functions. Well, now we had the hypothesis that the C-terminus inhibits, most likely by binding somehow to the transporter itself. And we remembered our earlier work done in the 1990s, 30 years ago, in which we had a similar mechanism for CSC2, for a plasma membrane channel. And there we know that the N-terminus of the protein binds to an intracellular loop of the transporter of the same molecule, the so-called JK linker between two transmembrane domains J and K. So we did mutations in the JK linker of the CSC3 to see whether we can affect the inhibition. So indeed, when we change in the chimeric molecule the JK linker of CSC3, and then we get the large vesicles again, but these are no longer inhibited by T9A or T9B, suggesting that the C-terminus binds in the vicinity of this intracellular loop. And interestingly, in this JK linker, there are human mutations in neurodegenerative disorders, which are shown here. This is for CSC4 with neurodegeneration. And we inserted these mutants into CSC3 and found that it partially disinhibits the inhibition. So this means that it activates the transporter, and this is a gain of function which is toxic for the patients, which really causes disease. And we looked at other mutations in CSC4. So this is again the transmembrane topology with the mutations known 2 years ago. Every year there's an addition. And there are several mutations which in heterologous expression do not give functional effects on the counts. So these green dots here are mutations which do not affect counts, but which cause disease. And since these are located intracellularly, cytoplasmically, we thought that these would be candidates for interacting residues with the T-man proteins, and we mutated all of them. In the meantime, we have got, in a collaboration, the cryo-EM structure, which is displayed here, and we found that 2 of these mutations are or mutants are no longer inhibited by TMEM9A. The other ones which are hidden in this intracellular domain do not do the trick, but these are again candidates for binding the C-terminus together with the JK linker, which is displayed above. We looked for more mutations described in human genetics papers, and we found that several mutations in human genetics affect the inhibition. They disinhibit the inhibition, they activate the channel. Not only in the CBS domain, but also in the amino terminus, the tyrosine, in another intracellular loop, and these two in the amino terminus. So this led us to speculate that this is kind of groove into which the C-terminus of the beta subunit binds and blocks the channel. Furthermore, we noticed that this residue and this other residue here are phosphorylated in databases. So this suggests that you can modulate the inhibition by the C-terminus by phosphorylation. And of course, we mutated these residues to glutamate to mimic phosphorylation, and we saw that the inhibition is partially gone. And we also saw that a particular serine in the C-terminus of the beta subunit of the TMEM9 is phosphorylated in databases. We did the mutation, and the inhibition was partially gone. So this strongly suggests that you can modulate the inhibition by the beta subunit, that is the activity of the CSC, by phosphorylation, which would be totally new. Well, this is a summary of the results so far. So we have identified in the C-terminus two motifs, the phosphorylated acidic cluster, which we name PAC, which is responsible for the endocytosis of the complex. And when we make mutations in this motif, we get less at the plasma membrane, therefore less currents and more in endosomes. On the other hand, we have the C-terminus of the beta subunit, which somehow blocks the transporter activity. And when we make mutations either in the CSC or in the C-terminus, we inhibit this inhibition, and then the transporter gets overactive, and we get swelling of endosomes, and this leads to pathology in the mutations we have studied. Well, early on, we contacted a cryo-M group, Richard Hyde at the Sloan Kettering Institute in New York, whether he would be interested to collaborate with us, and he got the cryo-M structure together with the beta subunit. This is the alpha subunit. CSCs are always dimers of the same protein with two translocation pathways, and to each of the monomers you have the TMEM subunits binding, mostly via the transmembrane domain, but also extracellularly or luminarily, and he also saw the C-terminus binding to the protein here. And I have to tell you a little bit of story as a caveat. Initially, he didn't see the C-terminus, and we had results in the meantime that the C-terminus, when we have an epitope attached to it, so we had HA epitope-tagged, C-terminally tagged proteins, which we know did no longer inhibit. I told him, do you have any amino acids at the C-terminus still from the purification procedure by doing fusion proteins? And he said, yes. I advised, take these amino acids off, and then only he saw the C-terminus. So the binding really depends on the not adding an epitope there. But the most interesting thing of the structure is, of course, the C-terminus and how it inhibits the currents. And very excitingly, I think, he found that the C-terminus, shown in yellow here, blocks directly the entrance pathway for chloride. So it really goes to the pore, it's not a channel, but to the translocation pathway, and blocks it physically. And you see it here. This is the permeation pathway in green. This is the cytosolic entrance for chloride into the transporter. Without the beta subunit and with the beta subunit, you get a constriction here, and the transport is really blocked. And this is very similar to CSC2. I introduced very superficially CSC2, where we had this JK linker and the amino terminus blocking it. And 2 years ago, a cryo-EM structure really confirmed our studies from over 30 years ago that the C-terminus binds to the JK linker, that the N-terminus of CSC2 binds to the JK linker. This is the N-terminus of CSC2. And very similarly now, the C-terminus of the beta subunit binds there and blocks the permeation pathway. And these are the amino acids which are involved in the binding. So we have kind of convergence in evolution. In CSC3, we have the beta subunit, which blocks the chloride pathway with the C-terminus. And in CSC2, we have the amino terminus of the same protein blocking the permeation pathway, which I think is pretty cool. Well, in his studies, he saw also another density in the cryo-EM structure, which we didn't know about from our studies, namely PI3-5P2 binding here. And I think that is pretty interesting. It binds between the CSC and the C-terminus. So it kind of is sandwiched between the two proteins, and it is important for the binding of the C-terminus. So I think it's the first time, maybe there are other examples, but for me it's the only example that the PI3-5P2 really links two proteins together. And some of these interactions are shown here in more detail. Of course, we mutated the corresponding amino acids in the T-mem proteins, also in the transporter itself, and saw functional effects. So the functional effects we saw were with apilimode, which is an inhibitor of PIK5, which is the enzyme which generates PI3-5P2. So when you apply apilimode, PI3-5P2 goes down, and this activates transporters. Let me go like this. So we have CSC3 giving the large vesicles. The large vesicle formation is inhibited by the beta subunit, but when we have apilimode, you get the activation, so it's no longer inhibited when PI3-5P2 is lacking. And we see the same effect also for CSC5, where you get large vesicles with apilimode. And we now know that CSC5 needs the beta subunit to go to endosomes and to form large vesicles. So it seems we have two mechanisms to regulate the activity of the transporter: phosphorylation of the transporter itself and the C-terminus, and changes in PI3-5P2. And let me now compare our results from disease and site-directed mutagenesis, how it fits really to the cryo-EM structure. So this is again the picture I showed you previously with the mutations in human genetic mutations, which indicated to us that there is a binding pocket for the C-terminus. And when you compare it to the cryo-EM structure, it fits pretty well, even though the distance is a little bit too large. But it is known that the C-terminus can move during activation. It is involved in gating, so we think that there's kind of mobility during gating. And the interaction is not always of the human genetic mutations direct. For instance, these mutations here, which I've shown you, which gave us these effects and are mutated in humans, interact indirectly with PI3-5P2 via interposed structures and conformational changes. Well, this is just a few keywords, what I've told you. Team 9A and B are novel obligatory beta subunits. They're essential, the knockout is lethal. They're needed for the CSC protein stability. They determine the trafficking and inhibit the transport of these transporters. Endocytosis depends on this acidic cluster. The C-terminal inhibitory domain blocks really directly physically the pore. The binding of CYT, this inhibitory domain, depends on PI3-5P2. And importantly, tonic inhibition of CSC is necessary for the physiology. If it is disinhibited, vesicles swell and you get disease. And loss of T9-mediated inhibition causes toxic CSC overactivity, which explains the pathology of several human mutations in neurodegenerative disorders. And these are the people who did these studies, most prominently Rosa, who is here in the audience, and who presented a poster which is still there, and you can discuss with her really the details of this work. Graduate students from Russia, Victoria Vorobyeva, Indian postdoc, Samantha Carr, and another graduate student. We collaborated with Bernd Fackler from Freiburg University, who did the SPLU-native page mass spectroscopy analysis, identifying for the first time these beta subunits. And we collaborated with Richard Hyde at the Sloan Kettering in New York to get the cryo-EM structure. And with this, I would like to thank you in several different languages and would be happy to take questions.
Thanks so much for this wonderful, exciting talk, and maybe there's time for a couple of questions.
Yeah, it's very exciting talk. I feel like, biochemist, I consider myself a biochemist, and I really appreciate your work here. So technically, a couple of, one is why actually neuronal degeneration? Why other tissues are not affected? I'm just curious. So, and second, of course, is what is a kinase? If you think about physiological relevance, how? Yeah. That's two questions. Yeah.
Well, neurological disease, I always make the hand-waving explanations that neurons don't divide, and therefore when they accumulate some intracellular structures, lysosomal storage and so on, they are more affected than other organs. With these CSCs, with some mutations, when you have activating mutations, you get also lysosomal storage with CSC7 all over, and even organomegaly, because these vesicles are too large. So it depends a little bit on the particular transporter, and the particular reason why the nervous system is not really known, but I think it really depends on the stability of neurons over time that they don't divide. The kinases is a very important question which we are trying to address. It's not easy because there are many different kinases. There are some consensus sequences, but not very strong ones, and we are now establishing a system to acutely measure the effect of inhibition of these transporters in endosomes by optical means to stimulate and inhibit some kinases, but you are limited with the number of tools. So it's a difficult question, but biologically a very important one.
Thank you. So, thank you very much. Appreciate it.
Thank you. Thank you very much for your very nice presentation. Well, I had two questions, but I can put it in one. First part of my question is how you try to recapitulate your very nice structural data by doing molecular dynamics.
Yeah, we have not yet done it because it would be interesting to see maybe moving of the C-terminus.
Yes, that's what I'm talking about.
But the time you need for the moving is probably too large for molecular dynamics, where you are limited to some
Very
nanoseconds
short.
and not tens or hundreds of microseconds. For the ion permeation of the CSCs, there's a lot of molecular dynamics trying to explain the exchange and the conductance and so on. But for the interaction with the PAI35P2 and the C-terminus, I think it's too slow to really make it with molecular dynamics.
And second question was, maybe I missed it. Did you identify the kinase which modifies the C-terminus?
No, not yet. We are looking into that. That's tough.
So
there's some consensus sequence weak for pKa. We are trying pKa. There are tools around. But in initial experiments, it wasn't really unambiguous. Maybe some effects. we are trying to do that. And there's some work in the kidney on endosomes that the chloride conductors might be modulated by pKa, but this is inner work.
Okay, thank you.
Okay, then I see one more question, but probably the last one.
If I saw it right, the human phenotype of the gain of function and the loss of function mutations was quite similar. Does that mean that having either too little and too much gives the same phenotype, and what would that mean for potential therapy? Because I imagine that we could try to modulate the function.
Well, in many cases, the phenotype is the same. So with CC7, we get osteopetrosis both with gain of function and loss of function mutations, which just means that when you mess up the system, you are sick in a sense. With CSC7 and also CSC6, on the other hand, we have strongly activating mutations. And with CSC7, which normally gives osteopetrosis, lysosomal storage, with strong activating mutations, you get no osteopetrosis, which is not understood, and lysosomal swelling in all organs. So there can be really difference between strong activation and inhibition. Of course, it depends a little bit on the disease. So with CSC5 is on the X chromosome. So here you find many loss of function mutations because they have only one allele. And with the other ones, it's autosomal. And it's mostly in humans gain of function mutations which are found because recessive disease is more rare, of course. But in many cases, it's kind of similar. With CSC3 also, I showed you the neurodegeneration. There was a total loss of function with one of the patients and gain of function mutations in others. So it's neurodevelopmental disease. The symptoms are quite variable. So from mild to really severe.
Okay, thank you, Thomas, once again for presenting these fresh data here and for the wonderful talk. I think we now have to move on for the young scientists, and probably Dos will introduce, and I just want to apologize that Thomas, Rosa, and I will have to go to the Newark conference because he was invited speaker there as well, and we have to do some duties there. So it's not disrespectful for the young scientists. Unfortunately, we have to go.
Okay, thank you very much. I appreciate it. So. Great talk, thank you. We'll follow up on details, how you lyse plasma membranes. I love it, I love this work, yeah. And I think now we have to move on with our, continue our conference. And right now I think we'll have a moderator with Ainur will be moderator of the young scientist, or Almagulia. So we have a list. So it's, as I mentioned earlier, so it's really important part of the training of our young scientists.
Okay.
Work is work in the lab, but presenting and understanding what you do, and if you explain to us what you do, it means you know what you're doing. And with that, I think we'll have, 7 minutes for presentations and 3 minutes for questions, correct?
Yeah.
And we have judges here, so be sure that and we have a prize at the end of the day that some will have to, for best speaker. And thank you very much. Let's get started, and Amul, you'll introduce first speaker? First speaker, I would like to introduce them. Kuanysh Dossybayeva, PhD student, and you in Matthew Tanker's lab, yeah.
Good morning, ladies and gentlemen. I appreciate this opportunity to start today's exciting and expiring contest, and I would like to talk about our study on the analysis of the T cells in pediatric patients affected with the immune-mediated disorders, and with the specific focus on the double negative T cells. This work was done at the Nazarbayev University School of Medicine, and I'm a PhD student on biomedical sciences. So T cells are key players of the adaptive immunity, and most of them represent the TCR complex of the T cells represent lineage-specific markers CD4 or CD8, but few unconventional T cells lack both of those costimulatory molecules and do not express, so they called double negative T cells. And we can define them on the flow cytometry by the negative expression of the CD8 and CD4 markers. Specifically, TCR complex of the double negative T cells can be of two types, alpha beta or the gamma delta subtypes. So why the double negative T cells? They are rare minor subpopulation, but well known that in the autoimmune lymphoproliferative syndrome, the expansion of the double negative T cells is a diagnostic marker. According to our systematic literature review, very limited information is available on the homeostasis of the double negative T cells in pediatric patients. So, in our study, we decided to evaluate the homeostasis of the double negative T cells in pediatric autoimmune and lymphoproliferative diseases context. So we enrolled in our study 434 patients admitted to the University Medical Center in Astana, which is the clinical academic division of the Nazarbayev University School of Medicine. We enrolled patients admitted in the rheumatology program, oncohematological program. In the additional groups were the infectious patients, mostly with the bacterial pneumonias and the control patients. So we collected peripheral blood from the participants, isolated the peripheral blood mononuclear cells, and performed our analysis. We were specifically focused on the T cell subpopulations and minor unconventional T cell subpopulations like double negative T cells, and among them the alpha beta and gamma delta T cells. So this is shortly about the method, our gating strategy. This plot represents from the healthy adolescent patients. So firstly, we isolate from the peripheral blood mononuclear cells, we isolate the lymphocytes, among them CD3 positive cells, and inside the population of the CD3 positive cells, we isolated the CD4, CD8 positive T cells, and specifically double negative T cells, splitting them into alpha, beta, and gamma delta T cells. This slide summarizes our findings recent. So here we evaluated 3 parameters. We measured the percentage of the double negative T cells within the lymphocytes, then within the CD3 positive lymphocytes, and the absolute counts of the double negative T cells. We measured all the 3 groups, like rheumatic group consists of the juvenile idiopathic arthritis patients and non-juvenile idiopathic arthritis, other rheumatic diseases patients, and we compared them with the infections, oncological patients, and the controls. So the group of the juvenile rheumatic group showed elevation of the double negative T cells compared to other groups, which suggests that the expansion of the double negative T cells might be a reproducible feature of the auto-inflammatory processes. And to conclude, I would say that we observed in our study the elevation of the double negative T cells, and this might be a non-specific marker of the autoimmune processes in this context. Thank you for your attention.
Kuanysh, thank you very much. It's great start of young scientist competition, so maybe we have some questions from our expert panel. We have 3 minutes for questions.
All right, thank you very much for this talk and this work. So did you control for other bias in your results, like gender, or sex, or age,
Yes.
and such? Well, how many factors, additional factors did you control in your statistical analysis for?
Thank you very much for your question. Of course, many things stay beyond of this presentation. So we applied the age-stratified approach. We divided our participants in 3 age groups from 2 to 6 years, from 6 to 12 years, and 12, 18 years adolescents. And also we applied we followed the gender balance. However, some types of the diseases cannot be, like systemic lupus erythematosus were mostly female patients of the adolescent age. Oncological patients, here we are talking about the acute lymphoblastic leukemia. They are the youngest patients mostly were enrolled in this project because it is the epidemiological representation of these diseases depends on that.
So you did. So
yes, we did that. We also did the disease-specific stratification, so each group has, for instance, GI group consists of the 6 subtypes of the GIA and other rheumatic diseases, but we are working on it, yes.
Okay, thank you.
Thank you, Kuanysh. Because of its competition, seems to me we need to have more questions, so maybe some questions from other audience. Yeah, thank you.
The main limitation of the study, so we started with the double negative T cells. The term double negative T cells is an umbrella term, I would say, uniting the minor subpopulation of the T cells that can functionally differ inside the group. So the main difficulty was in terms of the detection is that we are detecting those cells by the negative expression of the CD4 and CD8 T cells. There is no specific marker for those category of the cells. Then, however, we applied the alpha, beta, and gamma delta antibodies to detect specific cells inside the group. Other limitation is that comparing the rheumatic patients with the inflammatory diseases patients and the lymphoproliferative diseases, because in case of the lymphoproliferation, another challenge is to define the healthy T cells in the blood. Regarding the infectious disease, there is also lymphocytosis, and we cannot exactly define whether we are able to discriminate the double negative T cells inside the subpopulations. Thank you.
Okay, and one last question. No questions? Okay, thank you very much, Kuanysh. Okay, I'm sorry. No. No, please. It's really important.
Is anything known about the interleukin or cytokine spectrum occurring in these double negative cells?
Yeah, yes, thank you for your question. Yes, a lot of known about the cytokine profile depends on the origin of the double negative T cell. So there is several concepts about the origin of the double negative T cells. So they might be just escape the thymic maturation and appear in the blood, but sometimes also they might appear as the loss of the CD8 markers because of the constant autoimmune constant immune activation, and their functions differ from the regulatory to cytotoxic functions and according cytokine profile.
Okay, Kuanysh, thank you very much.
Thank you very much for your attention.
Congratulations for the participation. And I would like to invite next speaker, Kuanysh Sarkytbayev.
Hello everyone. So I want to start with my presentation with a little joke. This was my old presentation when I figured out in the midnight that I will be a presenter today. So let's start what did happen the last 10 hours. So my name is Kuanysh. I'm a research assistant in the Laboratory of Bioinformatics and Systems Biology. Today I'm going to present you the one-year duration project on the integrative analysis of transcriptomic data of esophageal squamous cell carcinoma. So first of all, let me start with a little introduction. The esophageal cancer, it has two major subtypes, and the main one that we will cover today is the esophageal squamous cell carcinoma. So as you see, the prevalence of the ESCC is over the 90%, and actually this is one of the severe subtypes because the 5-year survival rate of this ESCC is less than 10%. So if you look at the epidemiology studies, we can see that from the global in 2012, the incidence rates for them among the men from the older countries, we see that unfortunately Kazakhstan is one of those whose population has a predisposition for this disease. And according to, as you see, the East Asia, Central Asia, and the East Africa. So according to one of the last epidemiological studies, we see that ESCC actually incidence rates is 11, but the mortality rate is much higher, and we see that it's top 10 cancers. And unfortunately, again, 5-year prevalence is 7 to 100,000 population. So what is the significance of the studies? What I want to highlight, why we decided to investigate this one. First of all, it's one of the aggressive studies, aggressive cancers, and actually it is, even though it's in top 10 cancers, it is one of the most understudied cancers. And the second one, since it has problems with screening methods, most of these cancer diagnoses at the advanced stages. And of course, as mentioned, it's because of the local population predisposition for the cancer. So let me show the workflow of the project. So for this project, 16 patients were recruited, and these patients were not treated previously with the chemotherapy and radiotherapy. They were assigned for the surgical resection. The tumor tissue were collected and stored in the biobank of the National Research of Oncology Center, and we sequenced this data for RNA-seq and microRNA-seq, and then we proceeded the downstream analysis. Especially, we tried to look out the profile and try to find out the networks of the microRNA along the encoding RNA and mRNA, and then we tried to validate this data with the global databases like TCGA. So first of all, I want to highlight that the RNA-seq data is already published in the last year by our group. So we used the main focus for today would be the microRNA, which is not published yet. So the most of the data today it is unpublished. So this is the pipeline that we used for the microRNA analysis. So just little introduction. The microRNA, as you know, the last year the Nobel laureates got the prize for the microRNA research. These two gentlemen actually, they published the same paper, similar paper in 1993. They figured out that these two, LIN14 and LIN4, they have the same phenotypes, but one of them didn't have the expression product as a protein, but it's just a sequence. So first of all, that was quite uncommon situation for beginning of the 1990s, and they had phone call, try to reassure each other that it's something that is true. So in here we see that the microRNA actually it has significant impact on the gene expression. So the microRNA-seq data, so these are all data that we have. I don't want to get into details. So what we get first, we profiled our data, try to identify the differential expressed microRNAs, and we built up networks. But we decided very quickly, we decided to look at the common ones, all those 16 patients that they share the same differential expressed microRNAs and mRNAs. So among those 175 microRNAs, actually, the 100 of them, according to the MIR cancer database, all of them are considered as oncogenic. And according to cancer miRNA census, it's about 43. And we identified those which previously were not highlighted in the publications. But what I have identified that most of these microRNAs targets the one gene, this DDX6. So I tried to look at the TCGA database, to try to find out any association with the survival rate among the ECC patients. Unfortunately, as you can see, in other cancer types, it has some strong association. When it's highly downregulated, there is a low surveillance. But in case of the esophageal cancer, there is no difference. So since I'm a data analytics also in specialization, I decided to look at the more deep. So here, a very good paper which says that we need to check the reproducibility of the data, even the TCGA, because most of those who use in the TCGA data, they are not reproducible. So what we do, for example, let's imagine that we had the fossil full of bones, the archaeologist came to build something, and if he will not sort the things properly, he might get something that is very, very uncommon. If he will start sorting and try to label them correctly, he can sort them by the species. And that's the same for data analytics. We have the raw data, there's a data analyst, he tried to clean up data, and then we get the good results. That's the same thing I applied in my research. So by doing this, I have identified that DDX6 actually is highly associated with the surveillance. The high downregulation of this gene associated with low surveillance of the esophageal squamous cell carcinoma. And there's this little description. So in conclusion, I'm just running out of time. So I didn't show up the main our research output, those microRNAs which we will present on our publication as new biomarkers that we have identified among those new 16 patients, and we confirmed them on the databases. And we built a new networks which previously not also shown up in the papers. And one of the main one is that there's the gene DDX6 actually, which previously was not shown any association with the cancer. And yeah, so the main outcome findings were an independent validation to ensure the reproducibility. So acknowledgement to my PI, Dr. Ulkbek-Kayirov, and my colleague Madina Abdullayeva, who is an equal contributor for this project. Yeah, thank you very much.
Kuanysh, thank you very much. Amazing presentation and questions.
Thank you very much for the presentation. Can you quickly explain how you came to the difference, or which classification you did to come to the difference of DDX6 with and without correlation?
You mean that why I decided to choose the DDX? You mean that why I decided to look at the DDX6? Yeah?
No, why first you found no association, and then you found a correlation between the survival rate in DDX6 because of a classification, but what kind of?
Yeah, exactly. That's the beauty part of my work, because those papers, I highlighted them, they publish in the Cell Press, and they have the citation over the hundreds, a few hundreds, and most people, actually, oncologists cite them. And I reach out them, I look at this data that they use it, and I see that there are differences. And I reach out the authors, they are from Yale professors, and there are the two colleagues. I reached out that you have some problems with these mistakes, and they replied me actually in 3 days, and the answer was very simple. When I used the data for my analysis, I directly connected for TCGA, the Cancer Genome Atlas. But in their case, they used the data from the other authors who tried to clean up the data. And here we see that those patients that have the similar ADA data, the overall survival days are different. And I highlighted them, tried to indicate them. because we scientists, we try to contribute the common sense and try to make reproducibility, etc. And another thing, first, that the overall survival days are different of the 3 patients. Another method is that they use, for example, there's RNA-seq, yeah, and they use this way. They use the just log2 change, and in my case, they normalize the data with just log2. In my case, I use the DSEQ1 VST. So in that case, it considers the variance of the whole sample. So as you know, the DSEQ VST is more accurate and most recommended by the other authors. So there's another problem that because some of those samples which are downregulated or upregulated, they are wrongly shuffled. So the main in the priority are they use the log2 change to normalize the data. It's a little bit inaccurate. And second one, they have the overall survival is a little bit incorrect according to the main database.
Thank you.
One more question.
Okay, are you planning to test whether your microRNAs really target DDX6?
Yeah, so actually, as I mentioned, there are 175 common microRNAs which have the similar expression among the 16 patients. They're either upregulated or downregulated. And 20 of them, actually, over 20 microRNAs targets them DDX6, but I need to highlight that the 6 microRNAs which has the log fold change 4, it is too high expression of the microRNA. They all them target DDX6. So that was actually my reason why I decided to check DDX6, because I tried to look at the literature, why they are not highlighting this one. I looked at all the literature. Yeah, so...
Wait. So
yeah, validation, yeah, I know your part is validation.
It's very important.
Yeah.
Depending on, okay, so if they all target 3' UTR,
yeah.
but depending on how close you are to the end of the message, then the probability either goes up or down. So if you find that most of your microRNAs target the 3' UTR, say 800 or 1,000 nucleotides downstream, that means that most likely they do not target it.
Yeah, the only way is to validate through the experiment. Yeah, the RT-PCR that we plan to do for the publication.
Okay, I have a question about your bones. Did I understand it well that some of your patients underwent chemotherapy or radiation therapy before they got the surgery? No. Oh. So this is all primary tumor material that has not been influenced by chemotherapy or radiation?
Yeah, exactly. So they didn't have the prior treatment with chemotherapy either or radiotherapy.
Okay.
Yeah, but that's not the case for the TCJ because there are samples highly heterogeneous.
So could that also influence the expression pattern of your microRNAs?
Yeah, sure. There is that's one of the limitations of the study because we had the 16 patients, and we have the majority of them were in the stage of the 3, but all of the patients were free of metastasis, and we had very few, very few of those who are representing the stage 1, stage 2, and stage 4. The majority of them stage 3. So as you see, the limited number of the patients are doesn't make the highly heterogeneous the sample. So this is one of the limitations actually. But that's.
Okay, thank you.
One more question.
Very interesting talk.
Thank you very much.
That's my understanding that you've been doing bulk tumor sequencing, RNA sequencing. And one of the crucial points in this type of studies is to ensure the tumor purity.
Yeah.
So what was your approach to guarantee that you are sequencing tumor, namely squamous cells? Was it purely computational effort or some very, very initial steps like microdissection or something have been done?
Thank you very much for your question. This gives me opportunity to extend my presentation further. So the bulk RNA, this is one of the limitations that in tumor tissue we might have the infiltrated immune cells, we might have the muscles, and all that thing is represented on the signaling pathways. So also, like we know that the cancer is multiclonal, it's highly heterogeneous. We might have the different signaling, and one of those reasons that I might, for example, in our sample that we took aliquot from that, it might have the different clones of it, and they have different pathways and etc. In this case, yeah, sure. Even like when people do the bulk RNA, since they take the not all tissue for the sequencing, they might miss some important things. For example, when we do the targeted therapy, people do the biopsy. Unfortunately, they miss the main part, like which receptor they should target, like with the target therapy. That's actually one of the big disadvantages. In our case, how we did it, your question, we cannot escape one. We only can see the whole picture because, for example, in our case, we see, for example, some muscle tissue. As you know, the surgeons, they try to make sure to take the extra tissue. And this way we get the signaling for the muscle tissue, smooth muscle tissue, for example, because it's quite unique for this one. If you will have the from other part of the tissue, we will not get this type of signaling. And by the functional enrichment analysis, we can see the pattern, how they signaling. But of course, as you mentioned, this should be like the first step. Bioinformatics is like give us the direction, but other further steps should be detail it more. Like we should also prepare like RT-PCR. We need to go to cell culturing and etc.
Okay, thank you very much, Kuanysh, and let me invite here next participants, Darina Mukhamejanova.
Good morning, everyone. My name is Darina Mukhamejanova. I'm a first-year undergraduate biology student, and I'm deeply honored and pleased to present our research on oncogene-dependent mitochondrial dynamics modulation with the use of therapeutic drugs. So let's start. Because of the time constraints, I have to be brief, but I love to talk about mitochondria for hours. So let's start with KRAS and BRAF. Who are they? Are they oncogenic cooperators or they oncogenic competitors? Located being one of the most prevalent oncogenic drivers in colorectal cancer and PDAC, they are located primarily in the same signaling pathway. KRAS, when mutated, is persistently loaded with GTP independent of the growth factor signaling. BRAF, on the other hand, when mutated, is locked in the active so-called monomeric conformation and doesn't need any signaling from the KRAS to initiate further phosphorylation of the effectors like MEK or ERK. So located within one signaling pathway, they demonstrate quite different phenotypic control over mitochondria. And what's going on with mitochondria in KRAS and BRAF? Mitochondria is a very dynamic organelle. It constantly fuses and fizzes, constantly moves depending on the need of the cell, even if the cell is a cancerous cell. And here's some main features of oncogenic control over mitochondria by KRAS and BRAF. The principle is very simple. They upregulate and phosphorylate DRP to increase further fission and also suppress mitochondrial biogenesis in case of the BRAF mutant cells. What is interesting that forcing mitochondria to fuse by agents or genetic perturbation actually suppresses the growth of KRAS mutant cells. And even more interesting that the knockout of OPA1 is prevented tumor development in KRAS mutant cells, but not in BRAF mutant cells, giving us a clue that this main fusion protein is somewhat more needed for KRAS mutant cells than BRAF. And let's us introduce our model cell lines. Here we have CCD-con, the normal normal colon cells without keratinization that demonstrate the normal epithelial kind of type of cell of the colon. Also, we have the wild-type KRAS and our mutant KRAS and BRAF models. I know those confocal images look quite interesting and I love them, but let's try to quantify what's going on. And out of dozens of parameters that we had, we decided to focus on those two main parameters: total branch length per volume and endpoints to junctions ratio. What's endpoints? Endpoints is quite a rear edge of the mitochondria, while junction is the point where mitochondria fuse or kind of start forming a network. And what we see here is that HCT116 in both parameters shows to be most reticulated, and it also shows to have the highest extent of branching per mitochondrial mass. So out of this analysis, we formed this kind of spectrum continuum where HT29 BRAF mutant cell line and surprisingly CCD-con are less reticulated, where RKO, SVW, 620 and 48 are intermediate, and most reticulated is HCT. I want to make a remark that reticulated doesn't mean filamentous or even tubular. It means that this kind of mitochondria are more prone to form networks. More prone to form networks and connect from rods, from mesh to rods. Let's go next. We were quite curious what's going on with fission and fusion machinery. And here, situation is again quite evident and consistent with the literature. KRAS and BRAF mutant cell lines upregulate DRP and also push phosphorylation of the DRP at the serine 616, which further pushes mitochondria towards fission. However, what was interesting for us is that both KRAS and BRAF mutant cell lines remain just enough fusion machinery to support its oncogenic needs. As you can see here, the fusion in KRAS and BRAF mutant cell lines, particularly OPA1 levels, are quite high, and also quite high MFN2 levels, which is the main outer membrane fusing protein. And whenever MFN2 is absent, we see MFN1 kicking in and taking its part because of its partial redundancy. And what's going on? The Warburg-Reis KRAS cells, they maintain relatively high mitochondrial mass, which in results overheats mitochondria with production of more ROS and leads to somewhat loss of the mitochondrial membrane potential. And here's our modulation. We were thinking, can we modulate this mitochondrial dynamics and push them towards fission and fusion and reshape or remodel mitochondrial metabolism and find the particular vulnerability for those cells? Both of the molecules, M1 and MDV, they are small molecules based on hydrozones, highly hydrophobic, and supposedly one of them has to promote fusion, which is M1 mitochondrial fusion promoter, and MDV is supposed to suppress DRP1 and therefore also promote fusion. But the question is about M1. Is it mitochondrial fusion promoter or mitochondrial fusion modulator, or it's mitochondrial metabolism modulator? Please keep this question in mind till the end of the presentation. So we were curious, can we push our already normal colon model, CCD-con, towards more fusion with those drugs? And here's again quantification, and let's go to our main parameter, which is endpoints to junctions ratio. And as you can see here, M1, the effect of M1 shows the most reticulated mitochondria. So indeed, treatment with M1 pushes already quite filamentous CCD cone into more elongation.
Darina, sorry, time is over. Just conclude.
So the main takeaway from both of this, the drugs show some effect on both our KRAS block and BRAF block. And finally, the sneak peek that I want to give is that M1 pushes KRAS-driven cells towards more phosphorylation, towards more proliferation, but not the BRAF. And a sneak peek into the metabolic vulnerability is that priming with M1 for cycles increased the number of apoptotic cells in ATA-DVC treatment in 24 hours, but without priming, the number of apoptotic cells was twice less than we noticed in primed cells. So here's the acknowledgements. Thank you very much for all your contributions, and I'm happy to take any questions.
Thank you very much. Please, questions? Everybody can ask questions, not only jury. Please. First question. Very exciting results, but unfortunately we are urged to be at 7 minutes. That's why, sorry for interrupting, but still, is this one of criteria for judging as well? Да. And also questions, 3 questions for everybody. 3 minutes in questions. Okay.
Just one question, please.
So, excuse me, since we couldn't appreciate, which were the conclusions of your study, the main conclusions?
The main conclusion of the study is treatment with M1 and MDV pushes mitochondria towards network formation in KRAS mutant cells, but not in BRAF mutant cells, and also boosts its proliferation upon M1 treatment.
One more question. Yes, we have time. Yeah, please. Mm-hmm.
Can you quickly explain what drives the difference between then KRAS and BRAF mutations in your result?
Sorry, could you please repeat your question?
So can you then explain, you saw a difference between KRAS and BRAF mutation. What's the difference then, or how is that caused?
So one of the 2 main theories we adhere to is the glycolytic status. BRAF cells are more glycolytic than KRAS. That's one of the theories why BRAF are so resistant towards fusion. And the second is the origin of the mutation. Especially in HTT116, the mutation at codon 13 lies not within the P loop, but next to it. So those cells remain some sort of residual OXPHOS. That's why the further fusion may benefit to their oncogenic needs as proliferation or tumor survival.
Question here. I'm not a specialist, of course, but just I know that because it's always cancer cells, yeah? You use cancer cell in culture, yeah? And as I know, most of these cancer cells are use glycolysis, yeah? Instead of aerobic phosphorylation. Mm-hmm. And actually, biogenesis of mitochondria is highly disrupted in these cancer cells, yeah? Just one question. Did you try to grow these cells in galactose?
Actually, thank you very much. It's an amazing question. It's one of our future directives to grow cells in a different types of medium, like medium glucose medium or galactose medium. However, I would like to note that even though those cells are so prone to Warburg effect, they don't simply cripple off mitochondria. Mitochondria still remain very important factors for producing intermediates that needed for cancer cells to further proliferate and grow. But thank you, it's one of our future studies, and we will be happy to investigate it.
Thank you.
Thank you very much. That means really, really interesting results and fantastic presentation.
Thank you, Darina. So I just would like to remind you that Darina is just our undergrad student, okay? Undergrads, okay?
Thank you very much. Thank you very much, Darina. And I would like to invite our next speaker, Madina Abdullayeva. You come from Almaty, yes? Okay.
So, okay, good morning. Warm greetings from Almaty city. And today I would like to share our story on how we integrated data from genetic susceptibility and air pollution exposure so to predict asthma, bronchial asthma risk in an urban Kazakh individuals. So let's start. First of all, small introduction. If you ever come to Almaty, there is a 1 in 200 chance that the person you meet has a bronchial asthma. So from these numbers, you see the prevalence of this chronic inflammatory disease that affects airways, particularly the bronchioli. So they might be tightened, and there will be excessive mucus production. Of course, and all of this makes breathing difficult. And why we focused on this area is because the Almaty city is also surrounded by mountains, so the air particles are being trapped inside, and that's really difficult to control over them. And the main sources of these emissions and pollutions come from transportation, energy enterprises, and the private sector. And the highest number of cars is around 600,000. And unfortunately, our city gained an unpleasant title. This year, it became one of the most polluted cities, among others. So here comes the main research goals. Mainly, we were focusing on identification of single nucleotide polymorphisms specific to bronchial asthma, but then we also applied the environmental risk factors. So we evaluated air pollution state in Almaty, mainly focused on these 3 pollutants: particulate matter with 2.5 micrometer diameter and with the 10 micrometer, and sulfur dioxide. Then, of course, to assess association of asthma-related SMPs and their interaction with these pollutants. Research design looks like this, that we recruited asthma in control patients, all in all 288 people. We took their blood genomic DNA, then genotyped in Infinium HTS assay. Parallelly, we also took made a selection of certain SMPs from genome-wide association studies data. Along with that, we are also taking data from monitoring of 15 available air quality stations that were across the city. For example, you see here, and we've been monitoring from October last year till the September of this year. And finally, the statistical side of our project is to make association analysis for these SMPs and make interaction analysis in a program called Twink. This is just a visual representation of our research workflow. We prepare a customized survey for all of these patients. And of course, the main part of this was to have information about their place of residence, and so to assign stations where they live. So within the area up to 5 kilometers. This is the iScan platform, Infinium HTS assay, where we made genotyping. And the main statistical methods that we used is to assess logistic regression under the additive model for these asthma-related SMPs. This is measured by odds ratio. And for interaction model, we adjusted for the IQR, or interquartile range, levels of these pollutants from stations. And all of them was processed in the program called Twink GLM. And first of all, of course, we have to identify what SMPs have the predisposition to increase the predisposition to bronchial asthma. And all in all, we conducted about 88 candidate SMPs. And here comes the results. On this table, you see that our cohort was well balanced for these main factors like sex, age, and gender, and keep in mind that they were all Kazakh individuals. But the main difference, there was a statistical difference between having allergy symptoms. And that can be explained by the way that most of the people, asthmatic patients, they have allergy, of course, and there is even a tendency that if you have allergy, it might even flow to asthma in the future. Now, on this graph, you see the monthly air pollution levels for all of the free pollutants, and we see that mostly the tendency, there is a prevalence in increasing of the levels during autumn, like October, November, and even right now, we may also identify this trend. And the trend goes down during summer, but for sulfur dioxide, it rises up. But you see, for sulfur dioxide, it's like only at the green level, which means it's safe level. However, for particulate matter, it goes higher, yeah, during autumn and winter seasons. And we also were curious about making a correlation matrix between all of these pollutants, when we found that there were similar dispersal patterns for particulate matters, but none of the kind of correlation between particulate matter and sulfur dioxide. And here comes the main results of association, where we have been focusing only on identification of SNPs that are related to bronchial asthma. And this is the volcano plot, which represents our odds ratio values, but in logarithmic value. And we see, on the left side, we see this. These are the SNPs, and here on the circles, we see the annotated genes. And it represents the protective site, while this red side represents the risk site. And we see that SNP located in Gazdermin B gene shows the protective effect. So if a person has this SNP, it might serve as a protective variant, while those in the middle, we can say that there is a typical risk, not higher risk for these SNPs. And we were curious now on interaction between this having SNP and having a predisposition, affected by increasing level of pollutants. So for particulate matter 2.5, we see that if you have a SNP in a non-coding area located, and also with the rising level of particulate matter, this pollutant, it might serve as a protective variant. While on we had nothing in the risky side, so all of them are located on the moderate, representing typical values.
Madina, у вас тоже время выходит, поэтому.
Okay, next for the PM10, we see also the similar pattern, no risk variants, but more again the protective variants for these genes. And for sulfur dioxide, we also see the cytokine, interleukin 13, serves a protective variant, but this time we see the one risk variant, which is all again located in non-coding area. So keep in mind this. And we come to the conclusion that the levels of these pollutants are mostly at high levels during autumn and winter seasons. And protective SNPs, they are located in these genes. They may attenuate the inflammatory response to pollution. And risk-associated SNPs were identified in long non-coding regions. This gives us insights that the nature of these non-coding genes might be studied deeply in the near future. We have to focus on that. And it might give also a new direction for personalized medicine, today, one of the themes of this conference. So lastly, I would like to say special thanks to my great laboratory team and the laboratory head, Altynova Nazım Kalixanovna. So thank you for your attention.
Thank you very much. Please, questions.
Thank you very much. Very interesting study. It is quite well known that asthma symptoms become stronger when humidity becomes lower. So I'm wondering what are seasonal changes in humidity in Almaty city.
Yeah. Thank you for your question.
Whether it correlates with this air pollution indices.
Yeah, of course. So you pointed it right. It also depends. Humidity, we can include it also as a risk factor and also to monitor it. But unfortunately, we've been not focusing on that. but we have to also keep in mind, I think, yeah. But we see that, for example, the trend goes down during autumn season, and the humidity actually goes up during summer season, but we don't see any increasing trends on there. Of course, because humidity might help to reduce the symptoms because of the air pollutants, they are not dispersed that much when the humidity is increased. So during autumn, we see the negative trend on that.
Okay, and if I may, one more question. You've selected 80-something SNPs for analysis, and I was wondering whether, I'm not asking whether you have applied, but whether, what is your opinion regarding the necessity of correction for multiple testing in your study?
Yeah.
From statistical point of view.
Yeah, of course. For this study, we have to make several multiple assessment, validation, and for that, we need to make several tests, of course. But now, as you said, we also can make after we got the results. We actually want to check whether they cross the level of genome-wide association level, and also make also Bonferroni corrections for that, so to make a validation of our results.
So,
thank you.
Do you have time for one more question? So very interesting. But so, what's your as main advice that people who have protective SNPs, they have to stay, others have to move out from Almaty? No? Okay. Yeah. But I'm trying to get, so are you sure that how deep is your coverage of SNPs? So because you did mention, is it did you do global search for SNPs, or we just targeted?
Yeah, that was very quick, so because I was short in time, but in that, before we collected genome-wide association studies. So it was about up to 100 studies that were made that were focused on bronchial asthma. And we also had a chip which contains about 600,000 SNPs. So we were just parallelly crossing the data so that we also ensure that what other scientists have done before us, we also want to look in our individuals.
So it's all novel SNPs, correct?
Yeah, yeah.
And do you have what is your favorite SNP and how it could be relevant to asthma?
How is it relevant to asthma?
Yeah, yeah, yeah.
Oh, okay. So mainly, around those 88 SNPs, there were the major focus was mainly in cytokines, like interleukin 13, interleukin 6, and much more others. So mainly they were, because they modulate the airway, right? Hyperreactivity, airway modeling, so that's why the major focus was on them. Thank you for your question.
Okay, thank you very much. We need to follow our session. Next speaker, Madiyar Nurgaziyev, please.
Hello everyone, my name is Madiyar Nurgaziyev, and I'm a researcher at the Laboratory of Microbiome National Laboratory Astana. And today I'm pleased to present our work titled Interactions between the gut microbiome and the immune system in the development of rheumatoid arthritis. The work was recently published in peer-reviewed journal, and it's also part of my PhD thesis. Yeah, next slide. So what is rheumatoid arthritis? It is a chronic autoimmune disease characterized by joint inflammation, systematic immune dysregulation, and also progressive damage. Yeah. So here you can see how healthy joints look and how joints with rheumatoid arthritis look. Increasing evidence suggests that the gut microbiome plays an important role in shaping immune responses in rheumatoid arthritis. And however, in many populations, including Central Asia, there is a limited data on how gut bacteria correlate with the immune markers in RA. So the primary goal of our study was to investigate how the microbiota composition interact with various immune and inflammation markers in RA. So initially we included 400 patients with and after the exclusions, and missing samples, only 190 of them were included. It is 77 RA patients and 130 controls. So fecal samples were collected for 16S RNA microbiome sequencing, and blood samples were collected for cytokines, chemokines, and immunoglobulins analysis on the Luminax XMAP platform. Here's some information about our statistical analysis. We use Python. So the average age of our patients was 44 years, and disease duration was 9 years, and most of the patients was RF and ACPA positive. RF and ACPA are autoantibodies found in patients with rheumatoid arthritis, and they're often associated with a more severe form of the disease. And there are also other important clinical indicators such as DAS28. It is a clinical score that measures disease activity. Lower scores mean remission, higher scores mean more active disease. First, we observed that overall microbial diversity was reduced in patients with rheumatoid arthritis, and specifically alpha diversity measured by Chow and Face PD indices was significantly lower, while beta diversity analysis confirmed clear differences between two groups, RA and healthy controls. At the compositional level, the microbiome of RA patients was enriched in families such as Lachnaspiraceae, Clostridiaceae, and Ruminococcaceae, while healthy controls showed higher abundance of Staphylococcaceae and among the most abundant genera in RA were Doria, Tizarella, and Selimonus, all belonging to the Lachnaspiraceae family. When looking at autoantibody status, we found distinct associations in RF positive form of RA. Taxa such as Eubacterium and Monoglobus were enriched, while Prevotella and Butyrostrimonas were linked to RF negative form of the disease. On the immune side, patients with higher disease activity showed elevated levels of pro-inflammatory cytokines, including MIG, IP10, interleukin-6, and TNF-alpha. Importantly, Lachnaspiraceae was correlated positively with interleukin-5, and Oscillospiraceae correlated negatively with interleukin-22. So finally, our metabolic pathway analysis revealed that RA patients had enrichment of sulfur oxidation and glutamate-glutamine biosynthesis pathways. And healthy controls, in contrast, showed higher activity in lactose and galactose degradation pathways. So, yeah, that just a representation of Oscillospiraceae, Lachnaspiraceae diversity. Yeah. So, in summary, rheumatoid arthritis is linked to distinct gut microbiome with reduced diversity, increased Lachnaspiraceae, like I said before, and decreased Oscillospiraceae. Interactions between microbes, cytokines, and pathways, especially interleukin-5 and Leibniz-Forschungsinstitut, may contribute to the RA development. So these signatures hold promise as biomarkers and potential targets for microbiome-based therapies in future. Yeah, thank you.
Thank you very much, Madiyar. We have 20 seconds more. Questions, please.
Thank you for presentation. How do your results compare to other microbiome profiling studies in other autoimmune diseases?
The results are they're same. Recently, I've submitted our study. It was a systematic review. So in this systematic review, I included all the studies which studied, investigated the results of gut microbiome and also immune. It was a gut microbiome and rheumatoid arthritis. Yeah. I also included our study there, and we had some similar result with other studies. Yeah.
Just a question from a non-specialist. Thank you. You see, there is a cause and consequence, yeah? Maybe change in microbiota is a consequence of some dysfunction of immune system. Because in your presentation, you say that microbiota could be a cause, but how you would distinguish between cause and consequence? Maybe microbiota is just consequence. So even if you change microbiota, you wouldn't treat the cause, you see? And second question, what about antibiotics? Whether antibiotics aggravate arthritis? Or thank you.
The first question was about the consequences, you said, yeah? We cannot say that this bacteria can directly lead to that immune response, but there is a trend, some of the bacteria increases, and also there is a increased immune reaction. So that probably might be the reason for that. So, and the second question was about antibiotics. As you know, antibiotics, they kill the bacteria, and in microbiome is definitely there is consequences also of using antibiotics, and
the way aggravate arthritis.
Yeah.
Patients with suffer arthritis doesn't sensitive to antibiotics, so there is no data. Do you have any data? I don't know.
Unfortunately, we
anybody
don't have
studied
such
with
data. Maybe Alma can answer.
I'm not a participant of this competition. Okay, thank you very much. Time is over, and I would like to invite. Thank you very much, Madiyar. Congratulations. Okay, and I would like to invite here Asem Nispekova.
Hello everyone, my name is Asem Nispekova. I am a young researcher from the Laboratory of Bioengineering and Regenerative Medicine, and the title of my project is Targeting Small GTPase CDC42 in Aged Adipose-Derived Mesenchymal Stem Cells for the Improvement of Effectiveness of Cell Therapy for Bone Regeneration. As we age, our bone mass gradually declines, which leads to the heightened fracture risk. And did you know that about 75% of hip, spine, and forearm fractures happen in individuals aged 65 and over. And one of the key reasons why elderly people have impaired bone regeneration is because there is a decrease in stem cell number, and the quality of stem cells also decrease as well. So why stem cells are important, especially mesenchymal stem cells, they are key cells that differentiate into osteoblasts and chondrocytes, which in turn form a new bone. And mesenchymal stem cell therapy is a promising avenue for the treatment of bone fractures and for bone regeneration. And in fact, there are already 13 completed studies that study the effect of MSCs in bone regeneration. And there are two sources of mesenchymal stem cells. One is autologous and the second is allogenic. And autologous stem cell source is more attractive because there is no requirement for tissue matching, and there is almost no immune reaction. However, as we age, the quality of MSCs also diminishes, the migratory capacities and proliferative capacities of the cell decrease as well. And there are several strategies to improve mesenchymal stem cell quality, and today I will be focused on CDC42 protein. It's a small GTPase protein that plays a role in actin remodeling, cell migration, and other key cellular functions. And studies on human MSCs show that as we age, the activity of CDC42 increases. And that's why this protein can be used as a target for improvement of cell, for bone regeneration. And our lab previously demonstrated that as we age, the activity of CDC42 protein increases, and if we use inhibitor of CDC42 casein, we can decrease as beta-galactase positive cells, which means that it decreases the number of senescent cells after the treatment. So casein, how it inhibits CDC42, it binds to the CDC42 and prevents GEF function, which exchange GTP to GTP. So we saw that if we could inhibit CDC42 activity, we could improve the effectiveness of cell therapy for bone fracture repair. So first we wanted to test CDC42 inhibitor and as well as silencing RNAs, and we saw a substantial decrease in active CDC42 levels with both treatments. Next, we isolated adipose-derived mesenchymal stem cells, sorted out them with immunomagnetic cell sorting. We used CD105 for marker for that. And next, we wanted to confirm that these are indeed mesenchymal stem cells. We used mesenchymal stem cell markers to test, both positive and negative markers, and we differentiated into 3 lineages to confirm that they are able to differentiate into adipocytes, chondrocytes, and osteocytes. So next we wanted to test how this treatment affects the proliferation of the aged ADMCs, and also how it affects the senescence. So we have a result that it increases the proliferation of aged adipose-derived mesenchymal stem cells, and also decreases the senescent markers P21 and P16. And also it decreases the number of beta-galactase positive cells as well. And next, effectin is a stress cell marker. So when we have increased effectin levels, it means that the cells are stressed, and the treatment with casein and silencing RNA decreased the affecting levels and in comparison to young ADMCs levels. So next, we wanted to test if the treatment improves migratory capacities of the cells. For that, we used two assays, migration assay, transfer migration assay, and also wound healing assay. And both our treatments improved the number of migrated cells in transfer migration assay, as well as the speed of wound closure was also increased in treatment groups. Next, we wanted to test if the treatment improves osteogenic differentiation into the bone cells. So for that, we used ALP. ALP is the marker of bone mineralization, and with our treatment, we were able to see improvement in ALP concentration after the treatment. Next, we wanted to see if the treatment will have effect on anti-osteoclastic effect on osteoclasts. So we applied the secretome derived from the ADMCs into the osteoclasts that were seated into the collagen surface. So when we saw that the treatment improves the anti-osteoclastic activity of ADMCs, which means that the osteoclast could not eat up the collagen surface. Next, we wanted to see if our findings, in vitro findings, will translate into in vivo. Yes, so we had 4 groups: old controls, old with MSCs, old with casein in our cRNA, and we were able to see that in our especially cRNA treated group, the bone mineral density was increased substantially. So we analyzed the bone density for after 4, 6, 16, and 24 weeks. So yes, and we can conclude that silencing and inhibition of CDC42 improves migratory capacities, enhances osteogenesis, and these results translate into in vivo.
Thank you very much, and we have 2 minutes for questions, please. Questions? Be more active, please. Yes, thank you.
I'm not a specialist, but the question, if you make autologous therapy, and you do some treat with cells with silencing, I don't know, whether there is some risk of cancer, if there are potential risk of these cells, if you reprogram them for osteogenesis, whether we would migrate properly and do not become, in cell therapy, it's always this problem, even for autologous, I guess. So you have some idea, maybe whether it's very safe, or are you sure?
So,
could be.
Silencing RNA is not a treatment that will have effect in a long period of time or forever. So the effect is very limited in time, first of all. And the second of all, MSCs mostly die after 1 week after we inject them into the fracture area. What mostly affects the bone regeneration, it's secretome from the MSCs, most likely. So that's why I don't think that it can lead to cancer.
Okay, thank you.
No, very interesting. Yeah, so one more question. I think it's a CDC42 story. Why CDC42? Because there are other raw proteins, RAC, raw, correct? Why CDC42? And is it truly a marker of aging? I mean, do you have any idea what's the mechanism behind, or is it tissue specific, or something like your input on CDC42? Yeah.
So yes, you're right. There are lots of raw GTPases that play a role in many cellular processes. I think I have the slide in the. Yes, it's here. And why CDC42? First of all, CDC42 plays a role in osteogenesis. So in osteogenic lineage, CDC42 is more important because it activates the Wnt signaling, which prevents beta-catenin degradation. And why is it important? Because it activates RUNX2, which is transcription factor for osteogenic differentiation. So I think that's why CDC42 is more important in bone tissue.
And now, can we go this, when you have level abundance of CDC42, is it Western blot or? No, aging, or this one?
Yes.
Young and old, so is it?
This one is Western blot, right?
Western blot, correct?
Yes, yes.
But like, is it from mouse tissue?
This is from rat.
Rat.
Yes.
But do you have this kind of experience in human samples?
Yes,
Yeah.
so it's just the part of the, this study was not done by us, it's a study by Checker, and they isolated MSCs from 63 donors, so it's like from humans. And as you can see, they have a very strong correlation with increase in activation of CDC42 protein.
You mean abundance or activity? Here, GTP levels, is it?
Yes, activity, in GTP-bound form.
So increase?
Yes, yes, yes.
But in your case, you're talking about abundance,
No, I'm talking about, here see GTP-bound levels.
GTP-bound,
yes,
okay.
Yes, yes.
That's okay, good, now explain. Okay, so the activation, GTP-bound form is increased.
Yes.
But protein level is the same.
Might be the same, yes, you can see here.
Okay, good, just to clarify. Thank you very much.
Ratio, yes, right. I forgot to mention because
Asim, thank you very much. Let me invite next speaker, Aiman Jalmukhambetova. Aiman Jalmukhambetova.
Hello everyone, my name is Aiman Jalmukhambetova. I am a master student at Nazarbayev University and my PI is Professor Dossybayeva. So the topic of our study is oxidative stress induces cortical stiffening and cytoplasmic remodeling in pre-apoptotic cancer cells. So while oxidative stress is a major topic of our study, here we use two oxidative drugs, which are arsenic trioxide or ATO and D enantiomer of vitamin C or DVC to induce suicidal ROS production inside of cells. And here I'm going to briefly mention the mechanism of action. It's basically when vitamin C enters the bloodstream, it is converted into dehydroascorbic acid. And because it is structurally resembles glucose, it can enter cells via the GLUT1 transporter. And inside of cells, it starts to increase intracellular ROS levels. This is followed by the ATO treatment, and together they induce loss of ROS, and cancer cells can die. So here in our study, we use two cancer cell lines, AK192, which is mouse pancreatic cancer, and HCT116, which is human colon cancer. So here you can see from the microscopic observation, together a combination of oxidative drugs induces more apoptotic events in the cells and alter the cell morphology. But we were more interested in the brown section of the cells here, which are pre-apoptotic cancer cells, which just enter apoptotic stage but have not died yet. And we wanted to see what is happening to their biophysical properties, because it has not been studied yet. And because ATO-DVC doesn't kill cells immediately, we wanted to see how their functional abilities are changed, such as, for example, cell migration. That's why we performed scratch assay, and here you can see that the cell migration of ATO-DVC-treated AK cells decreased by 6 times, and migration of HCT cells by 1.5 times. And you can also see the same effect from the representative images. And while we admit that cell migration was partially affected by decreased cell proliferation, we still thought that probably cell migration has something to do with cytoskeletal remodeling and maybe the rigidity of cells. That's why we performed atomic force microscopy, and by using it, we measured cortical rigidity of the cells. And here you can see in red that the rigidity of cancer cells under the oxidative drug treatments increases by 2 times in both cell lines. And from the high resolution AFM images, you can see that the AutoDVC treatment disrupts cell cytoskeleton and alters the morphology, making cells more rounded, which we cannot see in control or single drug treatments. And because we know now that cytoskeleton is indeed different, we started to think which part of cell cytoskeleton is affected. And you might know that cell cytoskeleton is composed of 3 main filaments: actin, microtubules, and intermediate filaments. So, but we decided to focus on actin because actin is one of the most abundant protein in the cell. And it is important to mention that actin can exist in 2 forms: G-actin monomers, and once they are polymerized, they are converted into very long, stable F-actin filaments. And these F-actin filaments make a dense network inside of cell. To see whether cytoskeleton and F-actin levels contribute to cell rigidity, we decided to fluorescently label F-actin in our cells and measure the total levels of F-actin with flow cytometry. So here you can see that in AK cells, F-actin levels under AutoDVC treatment increased, but this was not true for HCT cells because they were the same as in control. This means that our hypothesis was wrong. But we decided to think further and thought, what if F-actin polymerization happens, but it depends on what part of the cell it is happening in? Because during atomic force microscopy, what we measure is the apical cortex of the cell. So basically, we measure actin meshwork under the cell membrane. So we decided to inhibit F-actin polymerization by using latronculin B. It is a depolymerizing agent. And what we have seen is that when F-actin is blocked, there is no increase in stiffness, which means that, and here is the same. So it means that cortical stiffness depends on F-actin level polymerization on the top part of the cell, and it is independent of total F-actin levels. Which was interesting observation. And next, we knew that AutoDVC treatment increases ROS levels, and we thought like, oh, what if ROS can drive all these events that we see in the cell? And to prove that, we decided to quench or block ROS with NAC antioxidant. And here you can see that NAC itself is not toxic to cells. But what is actually doing is that here, when we add NAC pretreatment and then oxidative drugs, there is no increase in stiffness or any changes, which means that the ROS is a key driver of cytoskeletal remodeling and cell stiffening. And to further prove whether it is a universal mechanism or not, we decided to use classical ROS-inducing agents such as H2O2 and rotenone. Rotenone is mitochondrial 1 inhibitor. So here from AFM you can see that under the treatment with oxidative drugs, the rigidity of cells increases as well as F-actin levels increases. So this was very important. Why? Because previously there were many debates in the literature whether there is any link between ROS levels and rigidity of cells. And here are 2 main conclusions that we can make. First, oxidative stress increases the cell rigidity in pre-apoptotic cancer cells. Second is that cell rigidity depends on F-actin levels on the cell cortex, but it is independent of total F-actin levels. And also we can maybe think in the future that cell rigidity can be some kind of biomarker of whether, in what state of apoptosis cell is and what are intracellular ROS levels. And I would like, and if you want to know more about this study, you can read our recent paper. And I would like to acknowledge people who contributed to the study, our colleagues and PIs without whom this study would not be possible. So thank you very much. And if you have any questions, I will be happy to answer them.
Thank you very much, Aiman. Please, questions, 9 seconds more.
Very general question, very nice piece of work, congratulations. So how do you see the main therapeutic application of your findings, if there are any?
Yeah, thank you so much. So the first one is maybe when people collect some patients' cancer cells, they can look whether cells are rigid or maybe they are soft cells, cancer cells, and they can see, oh, maybe if they are rigid, that maybe the cells are apoptotic and maybe cell is dying. So it may be the marker of apoptotic stage, and second, the ROS levels inside of cells. So as we can see here, the ROS levels increase, that's why rigidity increases. So this can be application. And also it is very important chunk for biophysical part of biology, because this was like a big dilemma previously.
All right. So you mean you can help to assess the prognosis of the disease, potentially?
Yeah, that's true, because atomic force microscopy is a bit complicated, but if you use some high-tech flow cytometries, you can quickly analyze patient samples and look at the rigidity of cells and make prognosis of some states of the cell. Yeah, so this is how it can work.
All right, so it can be a new functional test.
Yeah, maybe.
Maybe. Okay, thank you very much.
Thank you.
Okay. One more question, Bart, you would like? Bart.
Thank you, very nice presentation. If I understood well, often the effects of ATO and vitamin C were only there when they are synergistic. Can you speculate on why that is?
Yeah, that's true. This is very interesting thing, and why it happens, because ATO and DVC alone, we're using them in a low dosages that are not toxic to cells, which you can see from the apoptotic assays, from the morphological observation. But together, they just boost each other's effect on mitochondrial ROS production, and that's why they result in such a synergistic effect. But we're using the same dosages as for the treatments alone.
And would a higher dose of vitamin C do then the same thing?
Actually, let me clarify about that, because vitamin C does some cool thing, because if you look, for example, for migration, vitamin C boosts the migration even higher than for control cells. So it is interesting because like vitamin C is an antioxidant, but here we're using 1 millimolar, and it exceeds like cytoplasmic levels, the usual levels. That's why we're already using quite high dosage of vitamin C. Yeah, thank you.
Okay, thank you. One small question, if somebody has. No questions. Okay, thank you very much, Aiman. And our final for our session of Young Scientist Contest talk, Bayansulu Ilyassova.
Hello, everyone. My name is Bayansulu Ilyassova. I'm from Professor Dossarbasov's laboratory. So today I would like to talk about the defining of the gene expression regulation required for survival of cancer cells under nutrient or serum starvation conditions. So first of all, I would like to start from the ribosomal biogenesis process. So whole ribosomal biogenesis process starts from the nucleolus, where three centers, main centers and components like fibrillar center, dense fibrillar component, and granular component, where this whole pre-ribosomal biogenesis occurs. So, pre-ribosomal complexes consist of intermediate pre-ribosomes and composed pre-ribosomes. We know that cancer cells like nutrients and growth factors, and in cell culture we use specific mediums that contain glucose, amino acids, and the fetal bovine serum. So our main question was what happens if we remove from the medium main components, fetal bovine serum or amino acids, and how it impacts on the ribosomal biogenesis process and the mTOR signaling. So also our main research goal also was that determining the activation mechanism of adaptation of cancer cells when we remove specific components like serum or amino acids by performing the pre-ribosomal, polysomal profiling, and of course transcriptomic studies. And after performing this, it will lead us to understanding the processes of cancer cell survival. So first of all, of course, we started from the morphology. And as you can see, we performed the cell counting and the cell size measurements using our equipment Multisizer 4E. So also we did the variability assays, and we saw that cells are not dead, but they just slightly stopped the proliferation. Then we tried to check the ribosomal, polysomal, and the pre-ribosomal profiling under the serum starvation conditions. And we found, it was unexpectedly for us, that this under serum starvation conditions, our polysomes were very high as control. After 48 hours, you can see that polysomes slightly decreased. But also in pre-ribosomal peaks, you can see that they are also high. That means that there is an active translation. Therefore, if there is an active protein translation, we started to check the mTOR activity under taurine-1 treatment. And you can see here that under minus serum condition, taurine-1 treatment decreases the polysomal peaks. And also our Western blot analysis showed that phosphophore-beta activity also disappeared. That means that it all depends on the mTOR. So then we did the RNA sequencing analysis, and then we started to do the transcriptomic analysis of whole our polysomes, total, and ATS. But today I would like to show only total RNA transcriptomic analysis, and we found that such pathways like cholesterol and sterol pathways are very upregulated in under minus serum conditions. So then in NDMB231 cells, you can see also the upregulation of cholesterol pathways and other pathways related to hypoxia, but we focused on the cholesterol pathways. Then, of course, after transcriptomic analysis, you know that we should do validation of our genes that was upregulated. And we did the qPCR analysis and the Western blot. And here you can see that our genes that that participate in the myeloid pathway, cholesterol pathway, they are upregulated. And you can see it in our qPCR analysis and Western blot analysis. So then, we also tried to do validation with simvastatin drug. Simvastatin drug, this is a drug that inhibits the cholesterol pathway. Therefore, we used this simvastatin drug in 20 micromolar concentration, and we saw that under minus serum condition, you can see that cells are apoptotic and they are dead. So then we tried to check ROS production, ROS regeneration under this specific condition, and we found that after 24 hours and the 48 hours, there is a ROS accumulation under these conditions. You can see the shift in the MPI. So our main conclusions is that we indicated that after serum deprivation, we saw the high protein synthesis and the high polysomal profiling. Then we detected that the pre-ribosomal peaks also was very high under this condition, and the high protein synthesis rate in this serum deprivation condition is related to mTOR signaling. And our transcriptomic analysis revealed that these sets of mRNAs that relate to cholesterol biogenesis synthesis pathways were substantially upregulated under this condition. And then we can conclude that this expression of genes, they are responsible for lipid biosynthesis for survival of cancer cells under this condition. So I would like to thank you, Professor Dossybayeva, and the Laboratory of Cell Growth Regulation, also Laboratory of Genomics and Personalized Medicine, and the Laboratory of Bioinformatics and Systems Biology. Thank you very much for your attention.
Thank you very much, Bayansulu. Please, questions? We have 19 seconds more.
Thank you for your clear presentation. You checked the effect of serum deprivation on cancer cells. Do your results compare with what you find in control cells? Is there like any publication on that, or did you do it yourself?
Yeah, nice question, thank you. So I tried in primary cells, also in MRC5 cells I used, but I have just some technical problems with these cells because they grow very slowly for polysomal profiling, and now I'm generating these cells to checking the polysomal profiling also. I did polysomal profiling with these cells, but also I didn't include in the presentation, but we did polysomal profiling also in HEC293T cells, and we saw that the polysomal peak dropped. Yes, I could include, but sorry, I didn't include it in the presentation because of time limit. But yes, we used HEC293T for this, and we saw clear decreasing of polysomal peaks. Yeah.
Okay, thank you.
Thank you. More questions? No? Okay. Thank you very much.
Thank you.
Thank you. Okay, finally we listen to all our presenters for Young Research Contest, and I don't know how we will organize. Maybe our jury will talk to each other, or you can make scores for everybody, and then you can make one average.
I think we created a school just for you to help how to, but I think you can join together and discuss and choose the best one, because you must choose only one, the best. Yeah. You can discuss, we created it just for you, to help you. But it's totally up to you to decide. Okay, thank you.
Yeah.
And I think we will be back.
While our judge is working, let's take a picture. Young scientists who presented, like, they're on the way уже. See the? Сфотографируемся, да? Давайте.
Asiem, Aiman, Madina, young presenters. We need you for the photo. Мы потом еще снимем. Все, бегом давайте. Madiyar. Не, все потом. У нас. So we have a 30-minute break. 30-minute break, and we have coffee break in upper atrium. Девочки, а кто куда ушел? Девочки, кто выступал? Напишите в группу. Ребят, найдите Madiyara. Madiyara, позовите оттуда. Фото для и Kuanysh. Kuanysh полиграфии. Давайте все вместе сейчас, потому что ребята разошлись. Быстро сфоткаемся, а то потом... Давайте. Вы же стоите уже. Они собираются, с ними потом еще сфотографируемся. Часть убежала.
Может, сфотографируемся, когда будут?
Ну да, давайте.
Ну ладно.
Нет, давайте.
Я думала, просто молодежь сейчас...
Молодежь, давайте. Нет, когда будем которые выступали, давайте сейчас.
Вы будете эти фотки скачивать. Можно пока фоткаться на трибуне, потому что у нас не было до этого. Говорю, девочек потом на трибуне можно пофотографировать. Все? Готово? Дарина. Кто есть? Потом все, кто будет.
Наши atrium in half an hour break.
на трибуне хорошие фотографии. Сфотографируйтесь за трибуной. Давайте, вам нужен маленький конечно. Банальный фотоаппарат. Вы снимали бы Конечно. Не особо, да?
Пусть мама приедет. Пусть мама приедет, у меня будет такой...
Давайте, блин, ну я ж не накрашена, это вы накрашенные, уложенные, это нет.
И, ну, я не смогла. Спасибо.
Нет, это который... Все меняется.
Давайте, девчонок, Че, вставайте. Девочки, ну на трибуну, ну хорошая фотография. Мы потом в драйве все скинем. Вот туда, нет, за микрофон вставайте. Да.
Because we had an absolute consensus. We don't have any problems. All of us.
Can you write me on WhatsApp? So
What do you like me to do?
I will write down because I will be preparing the certificate.
It was this one. She was the best. And the second one was, I don't know you don't care about the second, but it was this girl. I think her problem was the number of slides, and too many, and let me see my books.
Что вам сказать?
Ну, вот у этой у нее даже нечего сказать. А вот у вашей, да, мне что-то то есть она там что, она не вовремя закончила, что-то у нее там было в этом представлении?
Не-не-не, просто, может, на вопросы там долго у нее спрашивают.
Ну да, что-то вот у нее было все хорошо. Да, ясно, я понимаю. Но здесь она... Вот, и вообще в следующий раз, пожалуйста, уберите всякие упоминания в группы, еще показывают фотографии своих руководителей. Там, вот это же влияет на оценку. Конечно. Ну, не в нашем случае, но это очень влияет. В следующий раз все убрать. Должно быть исключительно ресерч, а ничего больше. Никаких благодарности руководителю, фотографии с руководителем. Это не надо. Нам, на нашей
когда конкурс,
да.
когда доклады...
Она знает.
Рахмет. Спасибо большое.
Center for Life Science. Space where science comes alive, pushing the boundaries. Center for Life Science, one of the top 2 Center for Life Science. Space where science comes alive, pushing the boundaries of knowledge, turning vision into reality. Center for Life Science, one of the top 2 research.
Нет, чтобы вот это вот тоже было видно.
Okay, I think we crossed noon time. Good afternoon. Hoş geldinizler. Thank you for being with us. I think it's third day of the conference. It's the final moment now, and I think it was very exciting and very productive 3 days. And I think we learned a lot, and we really appreciate all our distinguished speakers who flew from very, very long distances, but even our speakers from Kazakhstan as well. Few speakers came, but I think it was really productive and I can say successful event of National Laboratory Astana. And so you can, if you would like to learn more about some labs at Center for Life Sciences, we can arrange some visit to our labs at the Center for Life Sciences. So, we're still, our judges are still working on finalizing our poster session, reviewing poster session, and but meanwhile we can talk and discuss if any input from our speakers, or anyone can say anything, have any idea to make some announcement or any other information, we can quickly actually discuss, or any questions. So I think you have to be sure that most of you are flying this morning, correct? So, or early morning tomorrow. So, but tonight, so afternoon, after lunch, we'll have some touring of the city on a bus, 2-story bus, I think. That's so you can see the city, enjoy city, and then after that, of course, you'll have time, you have a whole evening to enjoy, and then start packing, I think, and get ready, catch up with your flights. Yeah, don't forget. And thank you very much again, as I said. One more thing, as mentioned yesterday, probably didn't pay attention, but we also have a program, national government program of Balashak, and they are actively, so it's purpose of this program is to train our young scientists from Kazakhstan for 1 year at any top labs in the world. And I think that's, if any, we just have to locate strong, very good student, like a master, PhD holder, young scientists who will be willing to work in your lab. And we can arrange, we can start make a connection and networking. And I think this program, I recommend it to our government to increase this program from 1 year to up to 3 years. And I think then you can have young scientists for postdoctoral training. I think it will be a good way we can establish. First of all, we get strong training of highly competitive training for our young scientists. At the same time, these scientists can later set up strong collaboration with your labs, and I think future is bright. I think that's what the government is trying to do, provide all the resources and, of course, funding to maintain competitive, establish and maintain competitive research programs in the country. That will transform, that's only science can transform and move us forward. That's government is committed, and we're committed to deliver it. With that, our judges ready or not?
Постером готовы, да, уже наши?
I can keep talking.
Или мы будем пока по устным, а потом.
So, or if anyone would like to say a few words, please welcome. I mean, any points or any inputs, or how can we improve? How can we do better? But I think this was a nice setup, and again, I think actually. I was mostly signing papers. I didn't do much, but I was impressed how much work was done with our organizers to make this event happen. Not just happen, but successfully carry out all this conference at that high level.
This thing, I think yesterday we didn't show our small movie.
Yeah,
now it's ready. We can, while waiting our results of poster session, we can show now this movie.
Do we have a movie?
Yeah.
Our movie, a Malay movie? Can we start running? But I have to make an announcement. Movie was made by
organizing committee.
Handmade. No, I'm just kidding. We recruited people.
And also, some of you maybe was interviewed during conference, and after that we will include all interviews and photos and etc. to make another longer movie. And all photos and movie will be shared with you, with all of you.
Okay. Are we ready? Movie in how many? 1, 2, 3.
Welcome to the Center for Life Sciences, a unique space where science comes alive, pushing the boundaries of knowledge and turning vision into reality. The Center for Life Sciences is one of the top 2 research centers representing the National Laboratory Astana at Nazarbayev University. Established in 2010 under the leadership of Professor Sarkytbayev with the strong support of Nazarbayev University, the Center for Life Sciences is a leading biomedical research center in the nation and Central Asia. The core of the center is a team of the outstanding scientists advancing science by implementing cutting-edge global technologies with the focus to deliver high-impact research to improving healthcare and enhancing well-being. National Laboratory Astana is led by Professor Dossybayeva, an internationally recognized expert in biology and molecular medicine, and developer of an innovative anti-cancer drug. Over the past 15 years, the center has hosted 10 international conferences dedicated to regenerative and genomic personalized medicine, healthy aging, and sustainable health, alongside numerous workshops, seminars, and roundtables. The center is also deeply engaged in global collaboration, partnering with leading scientific institutions and participating in international consortia, such as the Human Microbiome Project and the Open Biodiversity and Health Big Data Initiative. Members of the center's team also serve on editorial boards in prestigious international journals. The director of the center is Professor Aiman Jalmukhambetova, author of over 300 scientific publications, and principal investigator of national and international projects in genomics and personalized medicine. The center proudly operates 8 state-of-the-art laboratories equipped with advanced high-tech equipment. Here, the scientists conduct both fundamental and applied research, leveraging cellular, genomic, and multi-omics technologies, model organisms, and cutting-edge approaches in systems biology and bioinformatics powered by artificial intelligence. In addition to discovery science, the center advances preclinical and clinical studies of novel therapeutics, while also designing innovative biosensors and bioinstruments. At its heart is a team of dedicated researchers, both domestic and international, many of whom trained at the world's leading universities. Their collective expertise drives forward Kazakhstan's biomedical research, with a mission to improve healthcare and enhance the quality of life to foster healthy longevity for future generations. The center is more than advanced equipment and cutting-edge technologies. It is, above all, about people. Here, ambitious young scientists, together with master's and PhD students, are shaping the science of the future. We are dedicated to nurturing the next generation of Kazakhstani researchers, providing them with world-class training and opportunities to grow. At the same time, the center remains a truly global hub, open to collaborations and partnerships with leading institutions around the world. The Center for Life Sciences is a place where innovation becomes discovery, and discovery becomes progress. Step by step, we are building a healthier future for all.
But we can start. Are we there yet, or? Very. But how about for presentations, for oral presentation? They said, what? Let's start with this. Что поговорить? I should come to this?
Да.
But we'll give right away?
Да.
Okay, good. Okay, meanwhile, so did you like the movie? No. Our acting was not good, but we tried, yeah. So I still have to talk because judges are working. There are many posters, and that's why, please, we still have time, correct? When this bus is, we have a lunch at 1, correct? 1:30, we have plenty of time. So, so let's continue, and if you have any questions, please don't be shy. You can, because we have time, and I think it's when we can actually maybe come up with some better ideas how we can network further, or because every university have different policies, correct? We can discuss our joint training program, or because we definitely have top students. One thing is things do not happen spontaneously, correct? Success of Nazarbayev University, it's built on the idea that we have to bring our talent to this university. That's why before there is whole system was developed by Ministry of Higher Education, Science and Higher Education. Before it was just Ministry of Education, correct, and Science. So there was national program was built on developing high schools, where top best talented high students were selected on after grade 7, I think, 6. So on grade 7 until grade 12, they were going to Nazarbayev intellectual schools, where program was more advanced, particularly in STEM sciences, social sciences, but also all teaching was done carried in English. So you can say that's why we could get top, and then that students from these schools, from Nazarbayev we call them niche, Nazarbayev intellectual schools, they were actually further selected to Nazarbayev University. And that's what I think we're getting top students, and that's why we're proud that we have talented students that we can teach them, we can interact with them, and they grow. So, I mean, I think we try to guide them and help them as much as we can, but that's why we have really a strong program, how we can move to the next stage. Because as you know, most important is to have top talent, okay? Human capital is #1 factor for success, because infrastructure comes secondary, but also important. Funding is also important, but without human capital, I think there is no chance to success. Okay, with that, I think we're getting close. Please, so Dr. Aiman Jalmukhambetova, please, you also be part of it. How we'll do it? Like, let's stay. Yeah, I think that. Yes, do we have to come here or better? So where is our cameraman? Cameraman is ready. Okay.
Maybe we will invite our committee.
Perfect.
For posters.
Who was the chair of our oral presentation committee? Please.
No, first maybe posters and then first posters.
Okay, you'll be ready.
Posters.
No, I'm just giving it to.
Yeah, for posters we created committee. It was 5 person. Yeah.
5, yeah.
Five person, leading researchers from different labs from our center, and they divided on 3 days posters, because posters, how many posters?
3 days.
48 posters.
48 posters, and they choose very carefully according to criteria, and now I think who will be announcing from results.
Why are you hiding? Come right here.
Who will tell results? You?
Okay.
Do you have enough microphones, please? 3 microphones. 3. No, it's okay. But they were not subjective, correct?
Yeah, we have.
No, it's not.
We choose only 3
Okay, very nice.
best posters, but it was really difficult because many of research it was very, very good, and anyway we make hard. We several hours make a decision who is best of the best, and probably Aidar Tairov for his research. It's #1.
First place?
Which place?
Of course we don't have, but by counting he's really first.
Let's take a picture.
This is 50K.
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 Let's take a picture all together. All together.
Сюда вставайте.
Cameraman.
Next poster, what we choose for the best, Altyn Zhuravlyova. Sorry.
Okay, Altyn Zhuravlyova getting compliment.
And again, 50K.
Very nice.
And final, Nurislan Mukhamejanova.
Нурислан Мухамеджанов.
Нурислан Мухамеджанов.
Он знает, да?
Нурислан Мухаmejanov.
$50K.
Thank you. Very good. It's a very good poster. We enjoy actually and discovered a lot of new for ourselves. And we know all directions. Many institutes really participate to our posters from NCBI, and also from Almaty, there was very, very good posters. And we want to give him them prize, but we limited amount of grants. Yeah. So next time maybe we can share it more.
They work better. Everybody improve. More posters. Just more posters. Yeah, it's okay. I think 3 awards are good for posters. I don't know, it's good. I mean, it's.
Okay, thank you.
So we have to go to the next.
All participants, yeah.
Now,
thank you.
Now it's a award for oral presentation. That's also next. And who announcement will be made by our scientist from Nagasaki, Vladimir Sayenko.
Dear colleagues, I am excited to announce that we've reached a verdict.
Very nice.
By our consensus opinion, the winner is Miss Aiman Jalmukhambetova.
Yes, Aiman, please.
Congratulations.
We were all intrigued to learn how does oxidative stress induces cortical stiffness in cancer cells. That was exciting. Congratulations.
Now you can hold it.
100K. You are undergrad or graduate? Master's student.
Thank you very much. That's my first time of presenting on a conference, and thank you for all your questions. Yeah.
Agata's was really instrumental in training Aiman. Huh? Was really very. Thank you very much, Agata. So, we wish all the best. I wish you all the best.
In Japanese, we say "gambatte kudasai," which means please work hard.
Kazakh say "Akhjo."
Thank you very much for our committee who making this decision. All posters or all presentations was very good, and hopefully next time we will do more. Not only 8, it will be more and more. Thank you very much.
Kob Rahmet, we really greatly your time is greatly appreciated. I think now I think was very intense time of all these 3 days, 3rd day. Now it's just relax, enjoy. Let's enjoy. Will be lunchtime for our speaker. Yeah, at 1:30, correct? And we wish you all the best and have a nice traveling. If I don't see someone, but. I feel we are just getting to the final point of this, our International Scientific Conference, Life Sciences Today, 2025.
One more thing, we would say thank you and gratitude to our organizing committee.
Oh, this is
and later for all our committee staff, we will give appreciation letters and maybe also some case. This will be, I mean, money. And it will be done by them, by a big team of this organizing committee. Thank you very much, headed by Yulia Safarova.
Yeah, let's give applause, great applause to our organizers of this big event. Yeah, it was, I think, went very well. Everything was taken care. I mean, all our speakers were actually met at the airport, brought to the hotels. I mean, it was a lot of logistics, but I mean, even organizing, setting up connection, and then talking to bringing our guests here was not simple. So I think it's serious work was done, and I think all this. Event, as many other things were brought together without any delay, serious delay, or some major problems. I think that was really successful. Due to our work of our organizing committee, and all work was led, again, I would like to say, Yulia Safarova, thank you very much. She did great, yeah. And we all recognize it, and I think we'll appreciate and we'll provide the award as well. Okay, with that, I would like to conclude today's scientific part of the conference, and I think now just enjoy your time, and I think, and get ready. I think we'll have also touring bus at what time?
2:30.
2:30.
2:30 will be bus.
After lunch at 2:30, bus, two-story bus will take you and drive around this, what's called, in the capital, Astana. You see, Astana is in I think it's a very nice city, particular.
If somebody would like to see laboratories, it's still we have time. Please tell us, and we will show our labs, and maybe no time before.
See, that's what it is. But nobody wants to leave, huh? Everybody wants to stay. Yeah, you want to go. So thank you.
If somebody wants to tell something.
Thank you. Thank you. Enjoy your time in Astana. You still have some few hours. Thank you for coming. And have a nice flight back, okay? Without any delay. I wish you good traveling back. Thank you.
Thank you for the organization.
Oh, yeah, thank you. That's Yulia. Thank you. Thank you very much.
We can still make photos in front of the banner outside. Yeah, outside, да, посмотрим? We have this banner outside. We can take a picture all together, yes? Ага. Девочки, фото. Можно, давайте, хотите групповое фото? Групп-фото. Давайте, да, здесь, вот тут свет есть. Ребят, групповое фото соберите всех вот сюда. Ну, всех, которые выходят.